And it's a pleasure to draw your attention now to feline coronavirus. And we have Kimberley Schmied from the IBI in Bern giving the first presentation.
Thank you very much. Hello everybody, I'm Kim. I'm a veterinarian and I'm doing my PhD in Volker Thiel's lab that you heard this morning as well. And I'm proof that there's also young people interested in, in veterinary coronaviruses as well. So I'm going to talk to you today about feline coronavirus. So probably you've heard about it, but if not, I'm just quickly going to show you why it's important. To put it into context, it's one of the alpha coronaviruses, so it's closely related to the transmissible gastroenteritis virus that you can find in pigs that causes a bad disease as well. Then in humans, it's close to the human coronavirus 229E, and in dogs, the canine coronavirus is closest related and plays also a role in feline coronaviruses. So why is it important and why is it interesting? I find it goes into both. So there's kind of, as you can see, two circles. We have one on this side and one on this side. And that's also, I always call it the evil end, that's this side, and the good twin of this virus. So let's get into it. Okay. So feline coronavirus circulates in the cats as the enteric coronavirus, so the FECV, and this infects the cats. It moves into the intestines and infects enterocytes, where it replicates and then it is shed via the feces to other cats. And this doesn't really cause much distress to the cat. It can cause mild enteritis, but usually nothing at all. Now, in some cats that are just very unfortunate, this virus then mutates and becomes the FIP virus, so the feline infectious peritonitis virus. We do not know exactly which changes and which mutations have to happen. There's been some guesses in the spike or ORF7, ORF3, but they do not explain the entirety of the situation. But what we do know is it changes tropism. So it goes into the monocytes, and in the monocytes, then it is distributed into all organs, so via the bloodstreams. And once it does that, it is very bad for the cat. So we're now on the evil twin side, and it causes a devastating disease in the end, killing the cat. And for a long time, we did not have any therapeutics against it. Then there popped up some therapeutics that were on the black market, so it was not a regulated treatment for veterinarians to give. But now this has changed. I know in the UK there's one that can be administered, and also in Switzerland we're getting there as well, which is really nice. So I think that makes the virus already very, very interesting, that we have these two pathotypes and this switch inside of the cat. But what makes it even more interesting, but also harder to research, is we also have two serotypes. So we start here with the FECV serotype 1, which is kind of the pure feline coronavirus. And then there's also serotype 2, where the canine coronavirus spike has kind of entered. So part of the genome is from the canine coronavirus. There's also some other differences, but that makes the major difference. Now, both the serotype 1 and the serotype 2 can be in either pathotype, so causing no disease as the enteric coronavirus, or then also as the FIP virus. Now, why am I saying that? It's A, interesting, B, serotype 1 is the one that circulates most in the cat population, so it's the one that is more prevalent. However, serotype 1 is also the one that does not like to grow in common cell culture systems, so we can go into the cat directly, but that's not ideal to study it. Or then there's also laboratory adapted strains, but that have many, many mutations compared to the ones circulating. Or then we have to come up with new systems to actually study serotype 1. Serotype 2 we can study in cell culture systems. So we thought we need a new system to study serotype 1, and that's what we did. So during my DVM, we had the idea that we would come up with a replicon system that we can put into BHK cells, so baby hamster kidney cells, because they're quite easily to manipulate and to handle. and then have this replicon RNA with a GFP reporter where we can test out different things like inhibitor tests. So here you can see what our basic idea is or was. So you have the entire FECV genome of a serotype 1, obviously, and you have these spike, membrane, envelope, and nucleocapsid structural genes. So we took out 3 of the 4. So spike, membrane, and envelope we kicked out and only kept in the nucleocapsid to have a replicating system that is then inside of the cells. And we also inserted a resistance cassette to select for it. And to make it visible as well, we added the GFP. So with this, we then have these cells where we have a replicating and translating virus or replicon, not entire virus, that cannot go out of the cells or infect new cells, but that we can visualize and test things on. And just to show that we managed to do that, you can see in green the cells. You can see also here some are a bit more green than others, but we have the replicon expressed, also the GFP expressed, and here we stained also for the nucleocapsid, so to really show it's not just the GFP that is made. Yeah, what did I do then with that? So my actual goal was to find some inhibitors that would inhibit the virus. So I took these replicon cells, these BHKF rep cells, and then I added different kinds of inhibitors and different concentrations. I incubated it and then did flow cytometry as a readout because I have the fluorescence. And then in parallel, I also, to show that the system really holds true, I infected, these are whole fetus 4 feline cells, and I infected them with a laboratory strain adapted FIP virus, also serotype 1, that has also a GFP inside. I did the exact same game, also with flow cytometry readout, as there's also a GFP. And in parallel, I also did TCID50 to check for actual viral titers, that it is really relatable to what I did with the replicon. What did I test? I tested remdesivir. You've most certainly heard of it as a COVID-19 treatment. It is an RDRP inhibitor and an adenosine analog, and the prodrug, the GS441524, is also has been used via the black market to treat cats that have FIP, and it's also now used in clinical trials as well. The advantage of the GS is it is orally available, so the cat can take it orally and then it is distributed well. However, the remdesivir has to be given IV. Anyways, this is also flowing into a clinical study at our university as well, where they're using both of them, remdesivir if the cat is really bad, and GS44 if the cat is doing better than to treat them, which is really nice. Then the GC376 has a different mode of action. It's a 3C-like protease inhibitor, and this one has also been shown in clinically experimentally infected cats to be effective against FIP virus. However, it's a little bit less potent than the remdesivir. Then nirmatrelvir is also a 3C-like protease inhibitor, the same as the 3C. And nirmatrelvir is part of Paxlovid, also a medication given for COVID-19 patients. And then amodiaquine was shown that it might have a synergistic or additive effect together with GC376. And in the beginning of the pandemic, there was also shown that it might have an effect on SARS-CoV-2, but I will get into that in a second. So here you can see, I will guide you through it. It's some big charts, but you always have on the left-hand side relative inhibition. So the circles, you want them to go down. That's good. And then the squares are the cell viability, so the right-hand line, and that's that you want to stay level. So if it goes down, the cells are dying. And then in the bottom, you have the different concentrations of inhibitor on a log scale. So remember that as well. So if it goes down, it goes up quite fast here with the concentrations. So as you can see, the GC376 as well as nirmatrelvir, so the two 3C-like protease inhibitors, they go down really nicely. The IC50 values are in a good range that we can achieve also most probably in vivo. then cell viability stays very level as well. So those work really nicely individually. And then remdesivir, it's similar. So we have a really nice inhibition. The IC50 value is also great. We can see that at some point it gets a bit cytotoxic. However, it's in a range that is really not problematic as the IC50 value is very, very low. For amodiaquine, as you can see here, the circles, they go up because the cells are dying here, and cell viability goes down. So amodiaquine alone did not work very well in our system. And they could show then also later on for SARS-CoV-2 that this was due, so in vitro, amodiaquine had the effect that during entry of the virus in the endosomes, it changed the pH, and that had the antiviral effect against SARS-CoV-2. And as you remember, I have a replicon system, so I do not have entry of the virus. That's why it also kind of makes sense now afterwards why it did not work. However, for SARS-CoV-2, they could show that in vivo, actually, this entry mechanism is not as important, so amodiaquine did not have a major effect in vivo, actually. That's why we didn't progress on that then also further. My idea was that it might be good to have combination therapeutics in the future, as then we can also lower the dosage and without have less side effects if that might be an issue. So we mixed the remdesivir with either one of the main protease inhibitors, so the 3C-like protease inhibitors. So you have in black the remdesivir by itself, and then in red combined with the GC376, and in blue combined with the nirmatrelvir. And you can see that the curve is shifted a bit below and a bit to the left, so it has some additive effect. However, it's not great, but it might still be a good thing to try out in the cats. And then if you combine both main protease inhibitors, so the GC and the nirmatrelvir, we can see there's pretty much a perfect additive effect. So this might be very interesting. I'm almost done. For the future. However, they bind to a similar location, so it might be that if it's not working for one, it might not work for the other, but it might still be very interesting. And in combination, we could show, I pushed wrong. In combination, we could show there was no additive effect with the amodiaquine. And as proof of concept, as I mentioned, I did it also with the laboratory strain adapted. And you can see here in black, it's the same again with flow cytometry readout for remdesivir and GC. And it looks pretty much the same as before with my replicon system. Also, the IC50 values overlay very nicely. And then in red, you can see the values for the TCID50 readouts. Also with wild virus, it overlays very nicely. So we could show that our system holds true. Now, just as a little outlook, what lies ahead, that was my work for my DVM. Now I'm in my PhD and I get into more detailed things, which is really nice. I'm currently trying to make feline intestinal organoids to really see where the virus binds, where it goes, what it does, in a more realistic setting, let's say, as it is also hard to study serotype 1 as a whole. Then I've been doing a lot lately on the single cycle variants. So with this, we would have the replicon system bringing in the rest and then have an easy exchangeable system where we can study different spikes and see what happens then. And as there was this outbreak in Cyprus of the feline coronavirus 23, we're currently also trying to make synthetic clone of feline coronavirus 23 to have a model to study as well. It's currently in the yeast for the TAR cloning, so I hope I will manage this soon. And with this, I'm at the end. I would like to thank the organizers for giving me the opportunity to talk to you about my projects. And thank you everybody for listening to me, and thank you everyone in the lab. It's amazing. I love it. And yeah, it's been a great experience.
Thank you. We have a bit of time for a couple of quick questions. Is there any way?
Great talk. I was just wondering about, so you say that for feline coronavirus 2, when it acquired the spike from canine coronavirus, and it still has that change to FIP, as does feline coronavirus 1. So what do we know about, because you say we don't know too much about what the actual virus changes is and what's dependent and what's required for the change. Do we know anything about what's driving the change in the host, like any host factors that are driving that?
We don't know exactly. We know that stress induces it. So if you have young cats that are infected already with the enteric virus, and you set them under a big stress like rehoming or traveling or whatever, it might induce more that FIP arises. So we know that there are some host factors that for sure play a role or pressurize it, but we don't know what exactly. No, it has not been studied, unfortunately.
Okay, thank you, Kimberley.
Thanks.